Welcome to all the students who are taking MOOC courses, especially the proteomics and proteogenomics course. Today we are going to talk about a very exciting technology, mass spectrometry, which has become the major working horse for the proteomic applications. As you've been already studying in your course, that how mass spec has revolutionized our ability to look at the complex proteome, its protein identification and quantification to a scale which was not possible earlier. Earlier, when we started our conventional 2D gel-based approaches, I hope you recall those techniques, where most of our, the major hits were coming from the gels with all these spots which we were seeing them on the 2D gels, right? But then moving forward, idea was, can we take the samples, whether it is coming from the tissue, body fluids, or any kind of biological sample, and rather than separating them on the gels, can we directly separate them with the different properties using mass spectrometry? So use different type of chromatographic principles followed by mass spec analysis. So that became shotgun proteomics approach. This entire trajectory of changing the field of proteomics, uh, reaching till the to the field of shotgun proteomics was all happening in the 1998 or 2000 kind of years. That's the time when 2D gel and the field of DIGE was getting developed. And then people realized how challenging it is to get the good quantification data from the gels. Because of the reproducibility issue that we have been discussing in the course, that's not very easy to do the high throughput proteomics on the gel. Then people realize, can we utilize the strong cation exchange and reverse phase chromatography, the two dimension of separation of the protein, and directly separate those chromatographic peaks and analyze them on the mass spec and use that information without gel. That was known as the first paper on the MUTPIT, multi-dimensional protein identification technology. And then field progress forward, because then idea was, we still want to quantify the signal. We want to still look at comparison of our control versus disease, and how to do all that if we are not running on the gel. Can we look at different condition, different quantification also on the directly mass spec based approach? But the current state of the art is that you can take any samples, including people are not even targeting single cell, to think about how applications can be done on the single cell level, especially when you think about the cancer tumor heterogeneity and many other biological applications where every cell itself is having a lot of its own property. And when you pool the different type of cells together, it may not give you meaningful data. So if one could take those any biological sample, directly extract the protein out of it, and those lysates are being digested to make the peptides. Especially you use the enzyme like trypsin, chymotrypsin, lysine, many type of enzymes you can use. And then followed by you separate them on the liquid chromatography, and then you want to do the mass spectrometry based analysis. This is quite important point which is now shown here in a recent review article by Matthias Munn team. There are two different approaches of now how you can uh, proceed for mass spec based analysis. Either you can use the DDA, data dependent acquisition, or you can use the DIA, data independent acquisition. So these two approaches conceptually have a lot of importance. For example, in DDA, you're looking at the most intense precursor, like what you're seeing in the red, the green, the blue. It's only picking up, the mass spec is only picking up these most intense precursors and trying to fragment those in a specific, millisecond time. And then goes to the next round and again picks up the most intense precursors and then try to further fragment those for MSMS data generation. So that is the DDA-based approach which has been conventionally used, whether we talk about our label-free quantification or we talk about TMT or iTrack, all of them works on this principle of looking at the top N precursors which are going to get fragmented. Then the newer approaches came, which is known as the DIA, data independent acquisition, where idea is, since we have peptides coming and eluting in different M by Z, starting let's say from 300 to 1300 M by Z, where most of our peptides will be seen. If we can take those small windows of 300 to 400, 4 to 500, and likewise we go to let's say 1300, And what you see now, those colors, like you are looking at every precursor in that M by Z, not only picking up the most intense ones, right? So you are now looking at the entire mass and charge window and trying to utilize that information irrespective of looking at only the top end. So this way, your coverage is becoming much broader. Of course, there's a lot of intricacies involved in doing DIA, whether need to do the library-free approach or library-based approach. How you're going to analyze your data becomes much more challenging in the DIA. Although there are newer approaches which are being utilized, software commercial like Spectronaut is there, but there are more free softwares like we have the DIAN, DIA DIAN software. We have the FragPipe. So some of these softwares have really started making the DIA analysis quite easy, and therefore your coverage can really jump up quite well. And the bias of the missing value, which we see more in the DDA data, where a lot of peptides are not getting sufficiently captured, those are known as missing values. That issue has been quite well resolved in the DIA. So these are the kind of, as I said, this is theoretical part. This can be quite important, and it has a lot of detail. Additionally, one could now look at validating your targets and do the targeted and NDIA, different type of analysis. Target proteomics itself is having a lot of interesting thing to talk, especially the two workflows of MRM, multiple reaction monitoring, and PRM, parallel reaction monitoring. And all of that we can try to look at a specific peptide, and we want to validate those peptides. So that we'll talk as we go along. Finally, then in this data, what comes forward is you get some sort of, the heat maps and the various ways of principal component analysis to find out differentiating your control and treatment, how well they can be differentiated, as you can see in this type of plots, where you in the volcano plot, you can see like, the proteins which are passing the threshold, which are extremely up or down, and significantly they're passing the threshold, those will be considered forward for your network analysis and the interaction analysis likewise. Yeah. So at each level, there are a lot of automations are happening, a lot of advancements are happening in the this new field of mass spectrometry-based proteomics, where a lot of robotics is being utilized to increase our throughput for the sample prep and looking at different ways of our speeding up the process and bring the more reproducibility. Different type of multiplexing possibilities are being also utilized, especially different type of chemical labeling approaches. Liquid chromatography is enriched with various type of new columns and different type of preform gradients. Especially EvoStep is one of the currently the big leader in the industry with advanced lot of our chromatographic experiences. And then there are different type of mass spectrometry instruments which are being used, which I will talk as we go along. So we don't want to make this again as a theory lecture and session right now. Idea was to give you uh, some update about the field and also show you live from the lab how different type of experiments can be performed. So TAs are going to take lead and they're going to show you different steps involved. So first we are going to talk about you have extracted the protein, how you're going to prepare the peptides. So if you were to do the gel, then you are doing the in-gel digestion. But in this case, we are going to avoid gels. So directly we are going to take the protein lysates in solution form and do the in-solution digestion. And once we have achieved that, then we would like to know how much quantity we have achieved from our peptide sample. The peptide quantification will perform. So both of these will be our hands-on session now, which we are going to do. Followed by, we will take these samples and do the mass spec analysis as we go along on different instruments. So I request now TAs to share the screen and proceed for the hands-on session on the peptide preparation in solution dilution. Amit, you are not audible. You have to unmute.
Hello, am I audible? Hello?
Start
Yeah,
again. Yes, now you're
yeah.
audible.
So good morning all. So welcome to the MOOC live session. I am a TA for the Introduction to Proteomics course. Today we are going to basically know about that how we can digest the proteins into peptides, digest the proteins into peptides, and that we can analyze for the mass spectrometry analysis. Now, now here you can see the lysates that we prepared in the last day. And now these are the proteins that are available in the lysates. Now the very first thing is that whenever you are going to start the digestion, you need to understand that how much protein that you need to first digest. So coming to the you can see Excel sheet here. So in the Excel sheet, you can see with the help of Bradford quantification, we have already measured that what is the concentration of the proteins that is present in the lysate. So today we are going to digest the protein sample that is present in this sample, UNDIL3. Now you can see this is the concentration. The 6.72 microgram per microliter is the protein concentration. And for digestion, we need almost 30 microgram of protein samples. Now, before starting the protein digestion, you need few important ingredients that will help in your digestion. The very first important thing that you need is the buffer B. So this is the buffer B. Actually, you need to make up the volume for the buffer B. Now you need to use the TCP. So TCP is a important gradient that will basically break the disulfide bonds. After TCP, you have the IA, the iodoacetamide. So this is the iodoacetamide you can see, and this is very light sensitive. You need to make them fresh whenever you are going to start the digestion. Now, what is the role of IA? So IA basically helps in the alkylation. It will prevent the reformation of the disulfide bonds. Now, and one of the most important ingredients is the trypsin. So trypsin, we all know that is the protease. It will basically break after the lysine and arginine. And question is why you use trypsin? There are also many proteases are there, right? We can break the long chain of proteins. So basically, trypsin will break in the arginine and lysine residue. One lysine is one of the most prominent or predominant amino acid that is widely present in all the protein molecules in the amino acid stretch. Now, the very first thing is that you can see that there are total 30 microgram of protein we need. So we will take the sample here. Now, you can see here the total 2.97 of microliter. That total 30 microgram. The 30 microgram protein is there. Now we will take only 3 microliter, 2.9 microliter of protein from the lysate. Yeah. Here we're putting the samples. Now we need to make the volume with the help of buffer B. Now buffer B, we need to make the volume up to 20 microliter. So 20 minus 3, that is 17 microliter of buffer B we need to put. We will keep the buffer B here. I will mix it well. And now you have the mixture of the buffer B and the lysate mixture. Now you need to add the TCP, which is basically useful for breaking the disulfide bond. So we take 1.2 microliter of TCP. And after mixing every component here, you need to show that you will vortex it well. So basically, for after adding every ingredient, there are some incubation time. Like even like one of the examples is that trypsin, which is caused by which we have to keep it for 37 degrees Celsius for overnight incubation. Now after adding the TCP, we need to keep it for always the 20 minutes of incubation. After we can add the IA. So IA is basically the compound that will prevent any kind of alkylation. So for right now, we will add the IA, and after that we will add the trypsin. trypsin to start the first round of digestion. So here we are using two different two-step digestion. Here we will add the trypsin for in 2 times, and we will keep the solution in 37 degrees Celsius. Now I'm going to add trypsin. And also after adding the trypsin, we need to also keep the solution, the pH of the solution 8. So for that, we will again need to add buffer B, and we need to check the pH. So actually the in solution digestion is quite a very long process. You need multiple ingredients, and you need to keep the solution in for 37 degrees Celsius incubation for a overnight incubation. Now, whenever your digestion is complete, you need to also acidify the solution with the help of Fa, the formic acid. And after with that, you need to first dissolve the entire solution. So in this entire procedure, we will use a lot of salts out there. So you need to also get rid of the salts. So with the help of these cartridges, you can get rid of the salts from the solvents or the solution. Now, after entire process, you will have the peptides here. You can see the very little amount of peptides here, and you need to add the Fa to again resolubilize the peptides. So for desolubilize, so after that, we need to find out that what is the amount of peptide concentration is there. So for that, you need almost two different kind of wavelengths. One is the 205 nanometer, and one is the 280 nanometer. So whenever you get the dried peptides, you need to resolubilize the pepper with the Fa, and after that, there is a particular micro droplet that I will show you. Yeah. So this is the micro droplet. You can see the peptides we need to put in this micro droplet, and uh, with the help of this instrument, we can easily find out that what is the amount of peptide out there. Now we are going to put the peptide solution inside the micro droplet. So I will be taking 2 microliter of samples. I'm going to keep it here. Simply again, I will be doing in replicate to minimize any kind of variation. Yeah. So again, before starting the experiment, we need to also set the blank here. So blank, we will use the Fa as a blank. I'm keeping the blank here. So you can see the drops at the micro droplet. Yeah. So now I will close it. I will close the lid. Yeah. So your micro droplet and your samples are ready. Now I'm going to share, and I'm going to show you a software which is called the Multiscan Go. So now we are going to keep the micro droplet inside the Multiscan Go. So you can see the plate is already out. I am keeping this microdroplet here. Now we are going to, you can see there is a plate layout here. Now we need to first set the plate template here. So this is a microdroplet. You can see thermoscientific microdroplet. I'll choose it. Then I have already put the two blanks here. So I'll keep blank in these two tabs. Then I'm going to understand, I'm going to find out what is the concentration of peptide. Now I'll keep set unknown here. I'm keeping in replicate, and I keep that in B. Okay. So now I have now set the plate layout. Now go to the protocol. Basically, in protocol, you need to set the absorbance. Absorbance, you will see use multiple wavelengths. 205 nanometers, and another wavelength is 280 nanometer. We'll keep precision. We'll go to absorbance. We'll perform blank subtraction, and then we will start the sample acquisition. Yeah. So after that, it will take few minutes, and it will start the peptide. We will get the absorbance of the peptides, and we will try to, and you can easily calculate that what is the amount of peptide concentration that is in our sample. Yeah. So you can see there total 1.494 is the absorbance, and 1.515 for the sample for the another replicate. So basically, you can export this entire information. So you have already this information. You can basically so this is the amount of 1.4 and 1.5 is the absorbance in the 205 nanometer, and 0.057 is the absorbance at 280 nanometer. So basically, you can keep this take these values and paste it in a particular Excel sheet. So basically, for this kind of peptide quantification, we use Scoop's law, where we use 205 nanometer and 280 nanometer and try to find what is the extraction coefficient of the 205 of the sample. Yeah. So now we'll basically drag it down. So there are total, you can see there are total the concentration of peptide is 0.6 microgram per microliter. And now if we talk about orbital fusion, for the in kind of global protease, we at least needs 1 microgram of peptide. For safer purpose, we generally give the machine to 3 microgram of protein peptide samples, and we also make up the volume with the help of FA. So thus, you will come to know that that's how so starting from Bradford quantification, you can see what is the amount of protein is there, and after using the trypsin and following some of the steps,